Okay, so hello everyone. It's my great pleasure to welcome you to the Global IMMUNOTALK 2023. I'm Kazuyo Moro from Osaka University, Graduate School of Medicine, Japan. Just before I introduce today's speaker, I would like to remind you next week's speaker is Marc Bajonel from CIML France. On behalf of all organizers of Global IMMUNOTALK, I would like to introduce today's speaker, Juliana Idoyaga. Juliana is an assistant professor in Department of Microbiology and Immunology at Stanford University School of Medicine, where she leads the Laboratory of Dendritic Cell Immunology. She received her BSc in Biology and Immunology from the Buenos Aires University in Argentina. And she then completed her PhD at the National Autonomous University of Mexico. And she performed her postdoctoral training at the Rockefeller University under the mentorship of Dr. Ralph Steinman. The goal of her research is to develop the next generation of therapeutic approach through a mechanistic understanding of DC. Her work so far has resulted in important discovery about DC subpopulation and tissue distribution and functional specialization, which impact on the basic DC biology and also development of the DC-based therapeutics. Her lab's ongoing research is focusing on the 4 fundamental areas of DC biology. First, origin and function of emerging DC subpopulation. And second, global transcriptional control of DC function. Third, functional specialization of tissue DC. And the last is DC contribution to the diverse immune responses in human. She's doing great work on establish new treatment for the cancer using DC, and I hope she's gonna talk about this today. So Juliana, could you turn on your camera for us?
Hello.
Hi, hello.
Great to see you. Thank you so much for an amazing introduction.
Thank you very much to joining to the Global IMMUNOTALK. We really looking forward to listening to your talk today. So as you know, we always start from one question from the speaker to speaker. And I heard you born in Argentina and then moved to Mexico. And now you are at Stanford University. So the question we love to know is about working abroad. So as an the network is not going well?
No, it didn't actually, yeah.
Okay, sorry, let's keep going. So you, I don't know where my connection start working.
With the when you were going to ask the question?
Okay, so the question is, as an immigrant yourself, what advice would you give to the young scientist who is training abroad is our question.
Okay, thank you very much. And let me first start by, thanking everyone at Global IMMUNOTALKs. First, because of the invitation, my real pleasure and honor to be part of this seminar series. And second, because of organizing this seminar series that really connects us immunologists around the globe. And I hope they continue going for many, many more years to come. So to your question, let me just first mention that, I do understand and I do know that being an immigrant, it does take a toll on us. It's hard to leave family, it's hard to live friends. But there is something very gratifying about being an immigrant and a scientist in another country or in another place, and that is we get exposed to diverse way of thinking, and that really pushes us to think differently. It pushes our brain to grow, and it really pushes our creativity. And so what I will advise to scientists that they are recently immigrated or they are thinking about immigrating, I will advise them to very, very fast when you arrive to that place to connect yourself with people, and connect with yourself with people that makes you feel safe and comfortable. And I think you shouldn't, you have to get out there. You shouldn't worry about language barriers, and just, build this web of people that I think is very important for your success as a person, as well as your scientific success. And hopefully you can establish long-term relationships across countries and across continents.
Thank you very much for excellent advice. I think people who wants to working abroad will get this advice very well. So now, could you share the
Yes,
slide?
of course.
And I'm a little bit afraid about the connection. In Japan, we had a typhoon today, and this is pre-recorded, so hopefully it works well. Okay, I can see
Okay.
your slide well.
Can you
And
see
if
my
you're
slides?
ready,
That's perfect. So give me a second.
okay, take your time.
Okay, that's great. Can you see my
Okay. Slides?
Yes?
Yes, sure.
That's fantastic.
Okay.
Okay, great.
Please.
So as you know, as Kazuyo mentioned, I am extremely excited about dendritic cells, and that is because dendritic cells. Oops, let me see why this is not working. There it is. So this is because dendritic cells, they are unique in their capacity to initiate, modulate a broad range of immune responses, from immunity to cancer, pathogens, even treatments, vaccines, to tolerance to self and environmental antigens. And that is why dendritic cells, they have a great therapeutic potential. And this therapeutic potential has been shown by so many groups around the globe, and using a lot of different mouse models. And in here, I would just like to exemplify that with my own work over the years in collaboration with, my mentors and colleagues. So early on in Argentina, I worked with Jose Mordo, with Rosa Weinstock, and Romina Goldsmith, who is now, as you may know, in the NIH. And we designed this strategy that basically consists in loading dendritic cells ex vivo with apoptotic tumor cells, and basically inoculating these cells into mice. And we show that this approach was able to basically promote CD4 and a CD8 T-cell immune response that promote the survival of tumor-bearing mice. Later on, with Can Yu at Rockefeller University, we decided to basically load dendritic cells in vivo with apoptotic tumor cells. And again, we showed that this approach was able to promote the survival of tumor-bearing mice. And later on, for my postdoc, I decided to join the lab of Ralph Steinman to take advantage of this approach that he designed together with Michelle Nusenswey, that basically deliver antigens to dendritic cells using anti-receptor monoclonal antibodies. And in collaboration with Hiroaki Hemi, we showed that this approach was able to protect mice from tumor challenges. In collaboration with Dinesh Matos, we showed that this approach was able to protect mice from parasite infections. And then later on, we decided to basically deliver to dendritic cells as self-antigens to promote regulatory T cells. And we showed that this approach was able to protect mice from an autoimmune disease, in this case, EAE, or experimental autoimmune encephalomyelitis. So as you can observe in this slide, basically these mouse models, they have demonstrated a great therapeutic potential. And the question is, what is happening in human. And unfortunately, harnessing human dendritic cells therapeutically has not achieved the promised potential. There is only one successful story, that is Provenge. It was a FDA approved in 2010 for the treatment of prostate cancer. So this approach was led by Ed Enkelman and Ron Levy, my colleagues here at Stanford, and consists on loading monocyte-derived dendritic cells with tumor antigens and reinfuse them into patients. But unfortunately, this approach, it only increased the average survival of patients by around 4 months, and there is no significant tumor size reduction. Moreover, this approach, it has been tested in other tumors and it hasn't been successful. So the question is why we cannot reproduce the success of our preclinical mouse data. And there is probably a lot of reasons why this is the case. But in the lab, we are working in two barriers that we think they are stopping our capacity to harness dendritic cells therapeutically. And the first barrier is that, as you know, dendritic cells are not one cell type. They are basically composed of subsets or subpopulations of dendritic cells. And we do need to understand the function of these subsets, especially those subsets localized in tissues. And we think that this is very important to harness these cells therapeutically. The second barrier that is basically the obvious one is humans are not mice. And there is evidence that dendritic cells function may differ between species. So for instance, the surface markers that we use to identify these cells, both in mouse and in human, are very different. And those surface markers are functional proteins. And so they are basically modulating function. So what we propose is that the dendritic cell network needs to be unraveled in the diverse human population and in tissues with the idea of finding parallels with mouse models. And we think this is very important to bring dendritic cells to therapeutics. Now, when we talk about the dendritic cell network, of course, we are talking about the presence of subsets. And I don't think subsets needs a lot of introduction for this audience, but I would like to briefly discuss them with you right now, so we are all in the same page. So you know that there are plasmacytoid dendritic cells, or pDCs, and there are conventional dendritic cells, the pDCs. They secrete a lot of type 1 interferon and respond to viral infection, where CDCs are very good at activating T-cell immune response. DC2, they activate CD4 T-cells, where DC1, they seem to be superior in their capacity to activate CD8 positive T-cells. Even they are superior capacity to cross-present cell-associated antigen. So the function, as well as the development of this subset, is of course determined by the action of transcription factors. And for today's talk, I would like you to remember this transcription factor, TCF4, also known as E22, that is important for the development as well as the function of plasmacytoid disease. And of course, these cells, they come from a progenitor cell localized in the bone marrow, a common DC progenitor that gives rise to basically these conventional dendritic cells. I'm going to call them the progenitor of conventional DC. I'm going to call pro-CDC. And the progenitor of plasmacytoid dendritic cells, I'm going to call pro-pDC. And in here, I would like to briefly mention that there is some, controversies regarding the origin of plasmacytoid disease, because it has been suggested that these cells, they mainly come from a lymphoid progenitor. And I will not discuss that today, but I would like to mention this manuscript by the group of Boris Raices. We contributed very, very little on this manuscript, but basically, what the group of Boris Raices, they show very elegantly is that plasmacytoid dendritic cells and CDCs, they do share a common dendritic cell progenitor that basically can be identified by the expression of CX3CR1. And so, this is, of course, it is a very simplified view of the dendritic cell network. And it has become even more apparent that this basically view of the dendritic cell network is simplified, I will say, in the last 5-7 years, because we started to use high dimensional approaches that allow us to observe that there is a lot of heterogeneity in the dendritic cell network that was not appreciated with the use of low dimensional approaches. And today, I would like to discuss with you about a subpopulation that we identify basically using these high-dimensional approaches. And this population I'm going to call transitional dendritic cells. And the work I'm going to mention is basically the work, it was started by my first postdoc, Marcela, and my first student, Becky, in the lab. This was a fantastic way to start the lab. These are fantastic trainees I had the pleasure to work with. And it has, the work I'm going to show you has continued to basically be done by Fernando and Raul in the lab, two fantastic postdocs that, again, I have the pleasure to work with. And I would like to mention that this work has been done very much in collaboration with a lot of people, but particularly with the lab of Boris Raíces. And so, as I mentioned, we started to basically characterize dendritic cells in human. This was done several years ago, not only in human blood, but also in tissues. And we chose at the beginning CyTOF. And we chose CyTOF for not only because it allowed us to, evaluate 40, 50 parameters at a single cell level, but importantly, because CyTOF doesn't have the problems of the autofluorescence that any other techniques, fluorescent techniques may have. And this is very important when we're talking about dendritic cells in tissues. And so when we basically analyze these dendritic cells by CyTOF, and this is in human blood, what we observe is this approach will allow us to, detect pDCs, DC2, DC1, but we also observe a population of cells that bridge between pDC and DC2. And because of this location in the maps, we started to call these cells transitional DCs. And it became very clear very fast that these cells, they basically localize to the same tissues as plasmacytoid dendritic cells. So transitional disease, we found them in blood, we found them in tonsils and spleen. But when actually we went to the skin where plasmacytoid dendritic cells are absent in healthy human skin, we observed that transitional dendritic cells, they were also absent. And that catch our attention because, it suggested that these cells, they may be related with pDCs. So in human blood, these transitional dendritic cells, they have very high levels of AXL. And actually, they were also observed by other groups. They were observed by the group of Aviv Regev with Mus Hanifa, with Bilani, with Chloe Bilani. And these cells, they were called as DCs because they express AXL and CD6 in human blood. They were also observed by the group of Florent Guignot, and for him, he called these cells pre-DCs, and I will discuss about that later on my talk. And actually, if you go back to the literature, there was observations of these cells back in 2009 by the group of Carolina Paluca, and they were described as a subpopulation of plasmacytoid dendritic cells expressing AXL. And what is very curious about these population of cells is that they really have a spectrum of phenotypes. So basically, their phenotype goes from a pDC phenotype to a CDC phenotype. So let me show you that. So here you have a pDC marker, CD123, highly expressing pDCs. You can observe that tDCs, they go from high to low expression of CD123. You look at CDC markers, CD33, CD11C, you see that these cells, they have low and high expression. And these are just some examples, but all the markers we have analyzed, we observe a pDC-like to a CDC2-like phenotype. And these heterogeneous phenotype basically correlates very nicely with the expression of the transcription factor TCF4. I mentioned TCF4, highly expressing pDCs, is necessary for pDC development and function. And you can observe that transitional dendritic cells, they have high and low levels of TCF4, depending on their pDC-like and DC2-like phenotype. And we know that TCF4 is active in these cells because we have performed ATAC-seq analysis. So we identified these cells in human, and then we started to wonder if these cells were present also in mice. So the first experiments we did were basically using cytoanalysis, in this case of mouse spleens. And what we observed again was that there was a population of cells that basically bridge between pDC and DC2. And they have high and low levels of TCF4. These are indeed transitional dendritic cells. When we analyze the marker expression, what we observe is that these transitional dendritic cells, they have a pDC-like to a DC2-like phenotype, and this is exemplified here with CD11C. You can observe high and low levels of CD11C. And because of this heterogeneous phenotype within the population, we started to divide the population in two, in particular based on their expression of CD11C, because that is conserved in mouse and human. And basically, we divided the cells in cells that they express low levels of CD11C. We call this CD11C low or tDC low. And those are more similar to plasmacytoid dendritic cells. And in cyan, here you can observe cells that they have basically higher expression of CD11C, that they are basically more similar to DC2, and we call this CD11C high or tDC. So this is phenotypic analysis. Of course, we evaluated the transcriptome both in mouse and in human, and we observed that indeed the mouse and the human tDCs are homologous transcriptionally. And we also observed that they are homologous in the expression of transcription factors. So here you have again TCF4 highly expressed in pDCs, and you can observe that transitional dendritic cells, they lose the expression of TCF4 when they start to resemble DC2. When we go to IRF8, another transcription factor, very nicely and very highly expressed in pDCs, it modulates plasmacytoid dendritic cell function. And you can observe again that transitional dendritic cells, they lose the expression of IRF8 when they start to resemble DC2. When you go to IRF4, a transcription factor that is very highly expressed in DC2. This is both in mouse and in human. In human, we observe that this transcription factor is also expressed by plasmacytoid dendritic cells. But either way, what we observe is in both the species, transitional dendritic cells, they express very high levels of IRF4. And finally, when we look at CDTB46, very highly expressed in CDCs, not in pDCs, and you can observe how these transitional dendritic cells, they acquire expression of CDTB46 when they start to resemble DC2. So basically, mouse and human tDCs are homologous not only in their phenotype, also in their transcriptome, and also in the expression of all of these transcription factors. So we were very surprised at the beginning that these cells, they are conserved, they are present in mouse, and we were surprised that no one actually observed them before, considering that the spleen is one of the organs that we study the most when we think about dendritic cells. And so we went back to the literature, and actually we found a manuscript published very early on, 2010, by the group of Stephen Young and Boris Raices, that describe basically a population of cells that they have high levels of T11C, high levels of CD8 and CXVCR1. And when we gated this population and overlaid them in our map of all the dendritic cell subsets, we found that indeed these cells that they name non-canonical dendritic cells, they fall within transitional dendritic cells, but they were only a part of those transitional dendritic cells. They were in a small part of those tDCs, and they mainly fall within this transitional dendritic cell high. So those means those tDCs that they are more similar to CDCs. And this was in 2010, but I would like to mention that in 2018, the group of Roxanne Tussin-Wan, she did observe a pDC-like cell. So this pDC-like, it has all the features of plasmacytoid dendritic cells, but they also express CBTB46. And actually, they seem to correspond to the tDC low, as recently described by Mark and Elena Tomasello. And so these cells, or at least a part of the spectrum, they were described in the literature. It's just that what I would like to emphasize in here is these cells, they're really on a spectrum of cells, that they go all the way from pDCs to DC2s. So there are cells that they are, they are very closely in phenotype to plasmacytosis dendritic cells, where other ones are closely to DC2 in phenotype. And in here, I would like to mention that, Chloe Villani, they suggest these are to be called as disease because in human they express AXL and CD6, but those markers are not conserved between species, and that is why we are proposing to call these cells transitional dendritic cells, so we can actually, emphasize this spectrum of phenotype within the population. And so I want to mention that these tDCs, they have been missed for decades. And probably because we've been using low dimensional analysis, and ergo, that means that these tDCs have been analyzed as contaminants of other DC populations. So those cells more similar to pDCs, they've been analyzed probably as pDCs. Cells more similar to DC2s, they have been analyzed as DC2s. And I mentioned that some of these transitional DCs, they express CD8, they express CD11C. So it's possible that in some experiments, they were analyzed as DC1. And for today's talk, what I really would like to discuss with you is two things. So first of all, I will discuss with you the origin of tDCs, and second, I would like to discuss the function of tDCs. So with respect to the origin, again, I mentioned that these transitional dendritic cells, they have a pDC-like and a DC2-like phenotype. So it makes sense for us to hypothesize that they could come from either pDCs from DC2 or from a totally different precursor. But very, very early on, when we observed these cells, we started to accumulate evidence that these transitional dendritic cells are developmentally related to pDCs. So let me walk you through that evidence. So first of all, we observe that they have lymphoid features, and that is a characteristic of pDCs. So here you have IgH rearrangements. pDCs, we can detect IgH rearrangements on them, but we can also detect them in tDCs, but not in CDCs. Another lymphoid feature we observe is that transitional dendritic cells similar to pDCs, they have PTCRA or pre-T cell receptor alpha, and that is not detected on CDCs. We also observed very recently that these cells, they actually, they say trace with every model that basically can trace plasmacytoid dendritic cells. So one of those models is the CD2 EYFP. So basically, this is a human CD2 crossed with a Rosa EYFP reporter mouse. And what you observe is that plasmacytoid dendritic cells are 100% labeled in this model. DC1s, we detect very, very little, if any, labeling. And you can observe that transitional dendritic cells we detect between 75 to even 90% in some cases. The other model I'm going to show you is a model that it was recently generated by the group of Boris Raíces, by Oriana Perez, and basically take advantage of the expression of CD300C by plasmacytoid dendritic cells. And in here, again, we observe that transitional dendritic cells, they're very nicely labeled. They're 75 to 80% labeled relative to plasmacytoid disease. And for today's talk, I would like you to remember this model, the CD2, because I will come back to it during my talk. And I would also like you to remember this result is that we do observe in both models a little bit of a staining in DC2. I will say between 25, 30% of a staining in those DC2s, and I will come back to that during my talk. And the final developmental feature that pDCs and tDCs share is the requirement of TCF4 for their development. So basically here we generated a mixed bone marrow chimera. And what we observe is that basically only in pDCs and in tDCs, the wild type bone marrow overcompetes the conditional knockout bone marrow. And that is not the case with CDCs. So basically, what I hope I was able to convey is that basically these transitional dendritic cells, both tDC low and tDC high, they have a lot of developmental features that they are sharing with plasmacytoid dendritic cells. So that is why we basically ask the obvious question, that is, do these transitional dendritic cells, they just originate from plasmacytoid dendritic cells at steady state? And we performed 2 experiments. So in the first experiment, what we did is we basically use a mouse model for the depletion of plasmacytoid dendritic cells. So that is a pDCA2 DTR that it was generated by the group of Marco Colonna. And in this model, you can observe that plasmacytoid DCs are very nicely depleted. And we asked what happened to the rest of the dendritic cells. And what we observed is that transitional dendritic cells are not depleted, suggesting that they are not coming from pDCs. Moreover, we decided to purify plasmacytoid dendritic cells and, adaptively transfer into congenic non-irradiated recipients and analyze what is their outcome over time. And we observed that when we purified pDCs from bone marrow, or when we purify pDCs from the spleen, what we observe is that pDCs, they maintain as pDCs after the adoptive transfer, and they do not become transitional dendritic cells. So with this, we concluded that tDCs are not derived from pDCs steady state. And so the question is, from where are these cells coming? And the dendritic cells, they all come from the bone marrow. So we went into the bone marrow and we asked if we could differentiate transitional dendritic cells from bone marrow progenitors using in vitro cultures. And indeed, what we observe is that cells that we from this culture, that cells that previously they were defined as plasmacytoid dendritic cells based on the expression of P220, PTCA1, and Siglec-H, they could be further divided into pDCs and transitional dendritic cells. And those transitional dendritic cells that we obtain from these in vitro cultures, they were very similar to their spleen counterparts, in meaning that they have this spectrum of phenotype from a pDC-like to a DC2-like cell. So that means that these tDCs, they can be obtained from bone marrow cultures. So the next question is, from where they're actually coming, which is the origin, which is this progenitor that is giving rise to these transitional dendritic cells. And I showed you at the beginning of the talk this slide that basically the conventional dendritic cells, or the CDCs, they derive from this pro-CDC expressed CD115, where the plasmacytoid dendritic cells, they seem to come from this pro-pDC CD115 negative precursor. So we ask, from where are tDCs originated, and importantly, if these tDC low and tDC high cells are related. One is more similar to plasmacytoid dendritic cell, where the other one is more similar to a DC2, so maybe they come from different progenitors. And so we did a lot of experiments to answer that question, and I'm just gonna summarize them in here. But basically what we observed when we performed single-cell RNA-seq analysis is that there was a cluster of cells in the bone marrow that it was able to basically bridge and give rise to plasmacytoid DC and tDC when we analyze it with trajectory analysis. And this cluster of cells actually corresponds to pro-pDC. So actually this pro-pDC seems to bridge and give rise to both cells. And to confirm that pro-pDC were actually able to give rise to transitional dendritic cells, we decided to purify them and to adaptively transfer them into a congenic non-irradiated recipient. And what we observed is indeed pro-pDC was able to give rise not only to plasmacytoid dendritic cells, but also to transitional dendritic cells. We also, we took a look at these transitional dendritic cells that we obtained over time after the adaptive transfer of pro-pDCs. And what we observed was very interesting. 2 days post-transfer, most of the transitional dendritic cells we actually recover. They were similar to plasmacytoid dendritic cells, so they were tDC low. Where 4 days post-transfer, most of the transitional dendritic cells that we recovered, they were actually tDC high, so that means that they were more similar to CDCs. So basically, these data suggest a transition from a pro-pDC to a tDC low to a tDC high. And this is actually summarizing here. What I mentioned is that these transitional dendritic cells, they actually seems to be derived from this pro-pDC, and we actually observe this transition from a tDC low to a tDC high. Now, in the next few seconds, I would like to discuss with you if these transitional dendritic cells, they contribute to the DC2 compartment. And this question comes because it was suggested very early on in human and very recently in mouse that these transitional disease actually can convert into a DC2. So we asked if this is the case, so we decided to basically purify these transitional dendritic cells and adoptively transfer them into a congenic non-irradiated recipient. And what we observe is indeed these transitional dendritic cells, they can convert into a DC2, and you have here the data. Very interesting. Early on, we observe also a population of cells that I'm going to call CD11B negative DC2. And basically, this population has all the markers of a DC2, but it lacks CD11B. And it seems to be this intermediate between a tDC and a DC2. So because these transitional dendritic cells, they can convert into a DC2, it has been suggested that these cells are just dendritic cells precursors or pre-DC2. And so we decided to evaluate if these transitional dendritic cells, they actually correspond to previous definitions of pre-DC2. And when we go to the spleen, we actually use definitions of pre-DC2 by Andreas Schlitzer when he was working with Florent Guignot. And basically, we gated pre-dendritic cells very similar to the way they gated back then. And we decided to overlay these cells into a UMAP of all the different dendritic cells populations. And what we observe is that pre-DC2s, they map, close by, but different than transitional dendritic cells. So really, they have a different phenotype that either tDC low, tDC high, or CD11B negative DC2. And we confirm this using this phase tracing model that I was talking about, the CD2 CRE EYFP, where indeed, as I mentioned before, pDCs are 100% labeled with this model. Transitional dendritic cells, we observe very nice labels similar to CD11B negative DC2, suggesting that again, all of these cells, they are related. But when you go to pre-DC2, you observe that these cells, they are actually not labeled with this model, suggesting that they are different, that tDCs are different from pre-DC2. And indeed, we also evaluated the turnover of all of these cells because it is very well known, it has been described that, dendritic cells, precursors, they basically have a very fast turnover. So we can evaluate that with a post-chase BRDU experiment. And this is the case, pre-DC2, they have a very fast turnover. They basically clear BRDU very fast, but when we go to the tDCs, we do not observe that. We observe that there is lower in clearing VRDU. So now I would like you to pay attention to the disease too. So I mentioned that transitional dendritic cells, they can actually, differentiate or they can convert into these disease too. So we started to wonder what type of DC2 they give rise to. And so, when we talk about DC2s, this is what I consider a bag of cells that they come probably from different places. And we know there is heterogeneity in this bag. And actually, it has been described in mouse spleen that this heterogeneity, and this is also functional heterogeneity, can be basically, dissociated by the use of ESAM and CXOCR1. And so basically, I'm gonna call these cells ESAM positive DC2s. They have also been called DC2A by Chrissy Brown with Rudensky, and these cells, basically the other subpopulation could be the CXVCR1 positive DC2s. That is how I'm gonna call them. They have also become called DC2B. These are pro-inflammatory DC2s. And so this is heterogeneity very nicely observed in the spleen. In other organs is more complicated, but in the spleen we can observe this heterogeneity. And what I want to mention is that it seems to be conserved between species. So it seems and it seems that the ESAM positive DC2 is equivalent to DC2 expressing CD5, where the CXVCR1 positive DC2s, they seem to be homologous to a CD14 positive DC3. And I do think that a little bit more of work is needed to basically confirm this homology between the subsets, but it seems that it is conserved between species, this heterogeneity among DC2. Either way, what we ask is if transitional dendritic cells were actually converting in either an ESAM positive DC2 or a CXVCR1 positive DC2. And the first thing we did is we just evaluated EYFP expression, and using that phage tracing I was talking about, the CD2 Cree EYFP mouse. And what we observed is that actually EYFP was only detected in ESAM positive DC2s and not in CXVCR1 positive DC2. Suggesting that tDCs could only contribute to ESAM positive DC2. And in here, I would like you to notice that not all the ESAM positive DC2s are actually labeled with CD2 EYFP, suggesting that there is further heterogeneity within those ESAM positive cells. So, do tDCs actually contribute to this ESAM positive DC2? And to answer that question, we decided to basically perform an adaptive transfer experiment in where we isolated the tDCs. We also isolated CD11B negative DC2s that I mentioned. They come from tDCs. We compare them with pre-DC2 as well as with pre-DC1. And as expected, pre-DC1 gives rise to DC1. So I'm not going to talk about these cells anymore. Pre-DC2s, they very fast and very efficient. They give rise to DC2s. And when we talk about tDCs, they also, they can convert into DC2s, but we see that they actually do that in steps. Actually, if we transfer CD11B negative DC2, we don't recover any tDC. And when we transfer tDC low, we recover some tDCs, but the recovery goes down over time, suggesting really a transition from a tDC low to a tDC high to a CD11B negative DC2. Either way, we wanted to analyze what is the phenotype of those DC2s that we recover after the adoptive transfer of these cells. And what we found is, as expected, when we actually transfer transitional dendritic cells or even CD11B negative DC2s, we do not obtain CXVCR1 positive DC2s. We only obtain cells that at least 70% of them, they express ESAM. And all of these cells, they express EYFP when we use the CD2 EYFP tracing model. But very interesting, when we transfer pre-DC2s, these cells, the DC2s we obtain, those cells, they also express ESAM. But those ESAM positive cells, they actually lack EYFP, suggesting again that this is two trajectories. One trajectory is a tDC, and it traces back to a plasmacytoid dendritic cell, and that is exactly what I mentioned, what I am saying here. So basically, these transitional dendritic cells, they trace back from a pro-pDC, and they can basically differentiate all the way to this ESAM positive DC2. And the other trajectory is basically these ESAM positive DC2s that they seem to come from pre-DC2, and that it traces back to this progenitor pro-CDC. And I don't have the time to show you, but I would like to mention that we have observed that human transitional dendritic cells, they can also convert into DC2s, but they only convert into these DC2s that they express CD5, and not the DC2s that they convert, that they express CD14. So that means that probably the CD5 positive DC2 is equivalent to this ESAM positive DC2. And we do not know if there is any functional specialization between these two types of DC2. However, because they have different origin, we are proposing that maybe it's a good idea to call them slightly different. So for example, if they come from a pro-CDC, maybe we should call them cDC2, but if they actually derive from a transitional dendritic cell, maybe we should call them tDC2. Either way, what we are sure is that these transitional dendritic cells, they do not differentiate in these CX3CR1 positive DC2. And if these CX3CR1 positive DC2s are the equivalent to the CD14 DC3, what we think is probably these cells, they come from a monocyte DC progenitor, as previously described in human, and they are not coming from either pro-CDC or from tDCs. So now in the last, a few minutes, I will shift, I would like very much to shift from, development to function of tDCs. And I show you that tDCs, they can convert into DC2s. However, we are convinced that these transitional dendritic cells, they have a lot of features of differentiated cells. And this was shown not only by us, by a lot of different groups. So for example, these transitional dendritic cells, they seem to promote allogenic T-cell proliferation. They quickly respond to TLR agonist stimulation. We have observed recently that they can uptake and process antigen for presentation to antigen-specific T cells. And it was recently observed very nicely that they can sense and respond to HIV. So we really think that these cells are not just, their fate is not just to convert into a DC2, that they really have function by themselves. And with respect to that, what I want to discuss with you is what is the in vivo function of transitional dendritic cells. We've been working for the longest time to try to figure it out, what they are doing in vivo. And the model we actually opted to use to evaluate the function of transitional dendritic cells was a murine coronavirus model. So this is also called a murine hepatitis virus. And we chose that model for several reasons. So first of all, we knew that tDCs, they were developmentally related to plasmacytoid dendritic cells. And we know that pDCs, they're very important during viral infection because they secrete type 1 interferon. And Luisa Cervantes very nicely showed that during the acute phase of murine coronavirus infection, plasmacytoid dendritic cells are key. They are very, very important to basically contain a virus. So we decided to basically infect mice with murine coronavirus. In this case, we inject mice IP, and the site of viral infection is the liver. And so when we look at the liver, what we observe is that very early on, so 2 days post-infection, transitional dendritic cells, they started to accumulate in that site of infection. So we ask, what is the function of those transitional dendritic cells that are accumulating? And, optimally, we will have a model for the depletion of transitional dendritic cells, but there is still, unfortunately, no model available. So what we decided to use is a combination of 2 different models. So we had a model that allows us to deplete plasmacytoid dendritic cells, as you can see here in red, but transitional dendritic cells are normal. And we have another model that allows us to deplete both transitional dendritic cells and plasmacytoid dendritic cells, as you can see here in blue. And I would like to stop for a second and mention that none of these models affected any other cell, or at least any other cell that we evaluated. We evaluated in a lymphocytes, monocytes, neutrophils, macrophages, other subset of dendritic cells, and I don't have the time to show you the data, but we observed that only these two populations, either pDC or pDC and tDC, are depleted. So we decided to evaluate what happened during murine coronavirus infection in these two depletion models. And let me just walk you through the data. So basically, when we infect mice with murine coronavirus, what happened is that a control mouse, they basically develop asymptomatic pathology. They survive the infection without any problem. But this survival is very much dependent on type 1 interferon receptor. When we use an interferon receptor knockout mice, the mice get extremely sick and they basically die by 2 days post-infection. So when we use a pDC depleted mouse, what happened is that we observed severe pathology. So that means that the mice started to lose weight, and by 5 days post-infection, all the mice, they have to be euthanized or they die. But when we use a pDC and tDC depleted mice, what we observe is a mild pathology. So that means that the mice, they did lose weight, but actually they survived the infection. And this difference in pathology was also observed in a lot of different, basically, parameters that we measure. So for example, the pDC depleted mice have a lot of liver damage as measured by high levels of the serum ALT 5 days post-infection. This is the day that actually the mice die. We also observe that these pDC depleted mice, remember in these mice, tDCs are there. What we observe is that there is a high infiltration of monocytes and neutrophils in these mice, and that is not the case when tDCs are absent. And finally, we even observe a spike in viral titers, meaning that those mice that they have severe pathology do not have pDCs. What we observe is viral titers is higher in the liver as well as the spleen. And I don't have the time to show you the data, but we observe this spread of virus to other organs. But that is not the case when we deplete transitional dendritic cells. So this data will suggest that basically these mice are getting sick. This severe pathology is induced because transitional dendritic cells are there. So we started to wonder, what are transitional dendritic cells doing to promote this severe pathology? And we perform these basically serum Luminex very early on after infection. This is 2 days post-infection. And what we observe is that in the pDC depleted mouse, there was a complete cytokine storm, but that was not the case when we also depleted transitional dendritic cells. And the cytokine that really caught our attention was actually IL-1 beta. And this cytokine catch our attention for several reasons. So first of all, these cytokines could actually explain all the cytokine storm that we were observing in blood. But also, it caught our attention because we had some data that when we activate with TLRs, agonists, transitional dendritic cells in vitro, we start to observe transcripts of IL-1 beta in those cells. So then we ask, do transitional dendritic cells actually produce IL-1 beta in response to murine coronavirus? And actually the answer is yes, they do. So we isolated those transitional dendritic cells and we incubate them with murine coronavirus. And indeed, we observe that they're able to produce IL-1 beta. And they actually produce higher levels of IL-1 beta than, even those ESAM positive DC2s that they convert into, showing that the capacity to produce IL-1 beta is actually in tDC itself and not in those DC2s they convert into. This is in mouse. The group of Boris Raices had this observation in where actually tDCs in human, they can also produce IL-1 beta. And this is against CPG, but they have also observed it in response to viral infection. And you can observe again here, human tDCs, they produce IL-1 beta and is at a higher levels than either pDCs or CDCs. So is this IL-1 beta produced by those transitional dendritic cells mediating all of this severe pathology during murine coronavirus when pDCs are not present? And to answer that question, what we did is we basically inoculated a single dose of anti-IL-1 beta antibody and evaluated what happened to pDC depleted mice, and actually we observed they survive much better infection. Moreover, they have a decrease in their liver damage. We observe decrease in serum ALT. We observe a decrease in this infiltration of monocytes and neutrophils into the tissue. So with all of this, let me just describe what is our current model of what is going on. So I mentioned these cells. They basically, they develop from a common progenitor in the bone marrow at steady state. And what happened is that during infection, these plasmacytosis disease, we know they produce a lot of type 1 interferon. And tDCs probably, they produce IL-1 beta, and they can also activate T cells. But what happened is that when pDCs are absent, these cells, they really get out of control. They produce a lot of IL-1 beta, and that promotes immune pathology to murine coronavirus. So in our current model, we would like to propose that both these tDC and pDCs are a lineage of dendritic cells with antiviral functions. And I hope with this, I was able to exemplify how learning about the subsets and their functions can hopefully bring us to therapeutics. And with this, I would like to close by thanking the people who works with me every day. I have an amazing team that I'm extremely proud about it. We have, I mentioned the members that they lead the work, but I also like to say that we are a team, we work as a team. And so in the lab, a lot of people has contributed to this work. I would like to mention David, Haley, Rebe, Andres, as well as past members of the lab. We have an amazing collaboration with a group of Boris Raices, and in particular, I would like to mention Oriana and DJ, who contributed to this work, as well as other members of Boris Lab. A lot of collaboration with people around Stanford, as well as I did in Mount Sinai. And yeah, I always like to thank, the donors, the surgeons, the pathologists, the nurses that help us to obtain that human samples, human tissues. And I would like to share with you that actually we were recruited very recently to the University of California in San Diego, and we will join them in August 2023. And we are very excited about, this again, moving and being exposed to a new community that is amazing. And so we are recruiting postdocs. So if you're interested, please email me and I will be happy to meet you. And with this, I want to thank you all for your attention.
Thank you, Juliana, for a wonderful talk. And your data was so beautiful, excellent talk. And I always get confused with like a DC subset because there are too many subsets, but now I can little bit understand, and I hope you can establish a good DC therapy soon. Thank you very much.
Thank you. Thank you. My pleasure. Thanks again for the invitation. Okay.
Thank you very much. And before closing, I would like to share one slide for people who wants to ask a question. Okay. And so please make a question through Twitter, as you know, and search for account Global IMMUNOTALK and find a Twitter that say ask question for Dr. Juliana Idoyaga here. So that's all for today, and good morning or good nice day, have a nice day or have a nice evening. I don't know, but that's all. Thank you very much for attending.